Welcome back, my dear students. Today we are going to explore the world of neurons and nervous tissue, the very foundation of our nervous system that controls everything we do from simple reflexes to complex thoughts. We'll see how neurons with their specialized structures like the cell body, dendrites and action work together to sense, transmit and process information. Alongside, we will understand the role of supporting cells, the neuroglia, and how neurons are functionally classified as sensory, motor and interneurons. By the end of this class, you will have a clear picture of how nervous tissue forms the structural and functional network that power our sensations, action, memories and responses. In short, how it makes us who we are. Nervous tissue components composed of two types of cells. Neurons are highly specialized, excitable cells that conduct impulses. Neuroglia, they are supporting cells that nourish and protect neurons. Functions: processing and transmission of information, connects all body regions with brain and spinal cord. It enables sensing, thinking, memory, muscle control and glandular regulation. This is the image of neuron showing the different components: dendrites, cell body, this is the nucleus within the cell body, this is action terminal, this is the node of Ranvier, this is myelin sheath, and within the myelin sheath there is one cell. Structure of a neuron: most neurons have three main parts: cell body, which is also known as soma or perikaryon. This is soma, or it is also known as perikaryon. It contains nucleus. This is nucleus and organelles like lysosomes, mitochondria, Golgi complex. It has also nasal bodies, which is made up of a rough endoplasmic reticulum with ribosomes. This is the sites of protein synthesis, repair and growth. It contains cytoskeleton, which includes neurofibrils that provide shape and support of the neuron soma. Microtubules, it transports various materials. There are also lipofunctin yellow-brown granules that accumulate with age. Nerve fiber, it is a general term for neuronal processes like dendrites and actions. Dendrites, they are the input regions. They receive signals from other cells. They are short, tapering, highly branched, tree-like structure. It contains nasal bodies, mitochondria and other cell organelles. Plasma membrane rich in receptor sites for neurotransmitter. In the plasma membrane of the dendrites, there are different types of receptor sites. Action: this is the action. This is single long thin projection arises from the action hillock. This is the action hillock. It triggered zone: action hillock plus initial segment. It is the site where the action potential arises. It contains mitochondria, microtubules, neurofibrils, but no RER or rough endoplasmic reticulum, so there is no protein synthesis. It has cytoplasm, exoplasm, plasma membrane or axolemma. Axon collaterals, it has side branches at right angles. It ends in action terminals or telodendria with synaptic end bulb or varicosities. It stores neurotransmitter in synaptic vesicles. Synapse and neurotransmitter, this is the image of synapse. It's the junction between two neurons. This is one neuron and this is another neuron. Two neurons, it can be two neuron or one neuron and another effector cell like muscle. So this is the neurotransmitter containing vesicle. So the neurotransmitter are the chemicals stored in synaptic vesicles. These are the round shaped structure, the spherical shaped structure, the vesicle, the electrochemical. So the neurotransmitter, the chemical stored within the synaptic vesicles, they excites or inhibits the postsynaptic cell. This is the postsynaptic cell and this is the presynaptic cell. A single neuron may release multiple different types of neurotransmitter. So you need to know actional transport. Neurons need transport of materials between soma and action terminals. Slow action transport, its rate is 1 to 5 millimeter per day one way from soma towards terminals. Move cytoplasm and structural protein, which is important in growth and regeneration. Fast axonal transport rate is 200 to 400 millimeter per day. It is a two-way movement along microtubules using motor proteins, anterograde or forward. soma to terminals, organelles and vesicles, and retrograde it is backward, terminals to soma, vesicles, worn out organelles, toxins or viruses. So this is the retrograde transport may carry NGF like the nerve growth factor or pathogens like rabies, polio, herpes, simplex, tetanus toxin, etc. Functional classification of neurons. This is the image showing the different types of neurons. First one is the sensory. This is the sensory or afferent neurons or unipolar neuron, you can say. It carry impulses from receptor to central nervous system. Receptors may be part of dendrites or separate cells, and basically they are mostly unipolar in nature. The second one is the motor or effector neuron. It carry impulses from the CNS to effectors or muscles and glands. They are found in cranial and spinal nerves. They are mostly multipolar in structure. These are the interneuron or association neuron. They are located within CNS. They connect sensory and motor neurons. They are responsible for integration and processing. They are mostly multipolar in structure. So in today's class we discovered how neurons and nervous tissue from the core of our nervous system. We saw how the cell body, dendrites and axon work together to transmit signal, how neuroglia support and protect neurons, and how neurons are functionally classified into sensory, motor and interneuron to carry out precise roles. This remarkable condition allow us to sense, think, move and respond to our environment. So always remember, behind every action, thought or memory lies the incredible network of neurons and nervous tissue working tirelessly. Keep revising, stay curious and see you in the next class. Thank you.